Welcome back! We've been learning all about the immune system, the things that it can do to prevent us from getting sick, and the things that it can do to help us when we do get sick. This comic is referenced by your textbook and is quite apropos to what we are going to talk about because sometimes the immune system doesn't behave the way it should. So Module 12 is all about diseases of the immune system. Sometimes the problem is in the microbes, sometimes the problem is us. So as we had discussed, the immune system is extremely powerful. It works wonderfully when it goes well, right? However, it's dangerous when things go wrong. So sometimes the immune system is referred to as this double-edged sword, with the idea that being, when it's attacking for you, it's your friend. When it's attacking against you, oh my goodness, so many different things that can go wrong. And we'll talk all about that in this module. There are many deficiencies, right? But generally it can be when you have an unwanted reaction, or sometimes the opposite would be that there's no reaction when there should be a reaction. Okay, so we're going to start off with talking about unwanted reactions first, and then we'll talk about what happens when there are no reactions. And the most common unwanted reaction can be called a hypersensitivity. And a hypersensitivity basically is just an undesirable immune reaction. There's some misconception here. We're thinking hypersensitive, that means, our immune system is super strong, right? That's really not the case here, right? So some of these things aren't necessarily your immune system is better. It's just doing what it's supposed to do against things that we don't want it to do, right? And so we'll talk about some of these mechanisms. We've already talked about some of these ideas behind hypersensitivity with respect to the good side of it, right? And we'll talk about how some of the things that our immune system does can lead to hypersensitivities. There are 4 types, cleverly named type 1, 2, 3, and 4. And each of these 4 types is based on a specific molecular mechanism, right? So even though they are all unwanted immune system reactions, their mechanisms are slightly different. So that's what I would like us to know. What is the mechanism of each of these hypersensitivities? Type 1 is all about IgE. Where's my pointer? It's all about IgE against soluble antigens, right? And remember, IgE attacks large parasites, but it also triggers inflammation, right, by binding to mast cells. So this is how type 1 works. Basically, we make IgE against some sort of antigen, and then it triggers mast cells, which leads to inflammation. And I would argue that all of us have had some sort of experience with a type 1 hypersensitivity. Maybe not all of us, but most of us, right? And we all know someone who does. And so let's stay tuned to that. Type 2 is all about IgG and IgM, right, against cell antigens. So here now we're attacking cells, right? But it's an antibody attack against cells. And we are pretty familiar with some of these things, and we'll talk about that a little bit later. Type 3, kind of rare. This is when antibodies form these things called immune complexes, right? And immune complexes are basically this clustering of antibodies up against tissues, and the body then has decided that, well, these antibodies clearly want to do something, and so they start attacking the tissue. Kind of a very specific situation. And sometimes it kind of plays within other diseases as well, right? But generally, it's I would say the rarest out of these types of hypersensitivities. I mean, that's what I would say. And then type 4 is cell-based, right? So type 1, 2, and 3 are all about antibodies doing the work, being the main culprit. Type 4, it's cell-based, activated macrophages, activated cytotoxic T lymphocytes, right? It's a T cell activated, T cell mediated tissue damage. And so we'll talk about how that works. Okay, so that's the basic snapshot. We'll talk about each of these hypersensitivities, some in a little bit more detail than others, and then we'll summarize towards the end of this, right? So your book actually has a really nice summary, but let's talk about each of these hypersensitivities and then summarize again. Starting with, let's start with type 1. Why not start with 1, right? So type 1 hypersensitivities are things that we actually refer to as allergies. So when you have allergies, right, this could be almost any type of allergy, right, including food, including pollen, including whatever, this would probably be a type 1 hypersensitivity. So the antigen, right, that you are reacting against, because remember, an antigen is simply something that stimulates the immune system, right? So technically it's an antigen, But in this case, we'll call it an allergen. So sometimes you'll hear it both ways, right? The antigen leading to type 1 hypersensitivity, or the allergen leading to type 1 hypersensitivity. And one of the common features of most of the hypersensitivities is this idea of sensitization, right? So actually, it's very rare that it's first exposure that leads to this hypersensitivity. It often takes some sort of sensitization. And so there are examples of this. We'll talk about this maybe on the next slide, right? This idea that it takes exposures to get there, right? So what happens then? First exposure activates our T helper 2 cells, right? Our Th2. And remember, Th2 are the ones that interact with B cells, right? And so what the activated Th2 cells do, so they bind the allergen, right? Or sorry, the allergen gets presented to T helper 2 cells, right? And so T helper 2 gets in there, and then it activates B cells. And it tells the B cells to make IgE. Right? And then these IgE eventually coat mast cells. And remember, mast cells are very important in inflammation. Right? So this is our sensitization process. We now have these mast cells that are coated in IgE, and they're primed and ready to release histamine or any type of inflammation mediator next time it sees the allergen. So subsequent exposure, and this would be the second to a millionth time that you see it, the allergen now binds to this IgE that's on the mast cells, and then histamine release, right? And then you get this inflammation, and then ultimately the inflammation that you feel really depends on where it's happening. And so we'll talk about that in a second. Okay, so it's probably easier to see this in a picture, right? So here we have our antigen, right? Remember, it's actually our allergen. So just for fun, let's call this some sort of pollen. I have hay fever, right? So this is what happened to me at one point. And this allergen gets taken up by this antigen presenting cell, right? Macrophage, dendritic cell, usually a dendritic cell. And then it shouldn't get processed because it's not really foreign, right? But for some reason, this antigen presenting cell decided that, you know what? I think I would like the immune system to check it out. And so it presents it on its surface on its MHC, right? So we learned about this in our previous module, right? And then so along comes our T helper 2 cell just doing what it does. It says, oh, you presented this to me. Clearly it's a bad thing, right? Activate it, and then help T helper 2 cells then activate our B cells, which then will differentiate, proliferate, and then differentiate into our plasma cells, right? So this is a nice review of how our immune system works, right? With respect to B cell activation, but then these B cells pump out IgE. Okay, IgE are, remember, they're inflammation mediators. So what they're going to do is they ultimately end up coating the mast cells. And then these mast cells are now primed and ready to bind this allergen again, right? So nothing has happened yet, right? A little bit of stuff, maybe tiny bit of inflammation, maybe, right? But generally now we have these mast cells that are coated with IgE. The next time, subsequent exposure, here comes our allergen again. This time now it's going to bind to the IgE. That's going to stimulate degranulation, right? The release of all those inflammatory mediators, and then we get inflammation, right? So a couple of key elements here. One, sensitization process, right? First exposure usually doesn't lead to a problem, and it's only subsequent exposure, right? And then the other is it's mediated by IgE, so it's antibody mediated, okay? So this is one of the reasons that you often develop allergies or things like hay fever later in life, right? Something like food allergies, oftentimes as a child, you never really had them, right? And you can kind of develop this over the course of your life, right? So for me, I didn't have hay fever until my freshman year, or maybe my senior year in high school, freshman year in college, something like that. And I just remember one day I just felt like I had a cold, and my nose was running like crazy, and so I thought, oh shoot, I have a cold. And then this cold seemed to go on for like weeks. I'm like, this is way too long for a cold, right? And so I said, maybe it's hay fever, right? So I took some antihistamines, some allergy meds, right? And then poof, just went away. I'm like, shoot, I guess I have hay fever now. That is my life, right? And so that's one of the things. So at some point, I must have become sensitized to whatever causing gave me hay fever, and then subsequent exposure in the years after now, I've become quite sensitive to that, right? So it's something that I deal with. It's not terrible, right? For some people, it's really bad, and we'll talk about what you can do when it's really bad. Okay, so that's the general reaction, right? The outcomes of the reaction really depends on the type of hypersensitivity that is induced. And generally, we can call them localized or systemic. Localized, as we know, means that it's restricted to a certain area, maybe a tissue, right? Systemic means whole body type of stuff, right? So, things like hay fever are considered localized type 1 hypersensitivities because it's limited to the respiratory tract, right? You can have some food allergies that are localized to the GI tract, often leading to things like hives, right? Some asthmas are type 1 hypersensitivities, right? A little bit more severe than hay fever, obviously, right? And then the outcome will, again, depend on the tissue, right? So, in secretions. So typically, if it's respiratory, you're going to have these secretions because it's going to try to get rid of it, right? You may get sneezing and that kind of stuff. If it's intestinal, you're going to get these hives. Sometimes you get cramping, all these kinds of stuff. And again, a little bit more severe, you may have difficulty breathing. The systemic type 1 hypersensitivity is quite dangerous. It's often called anaphylaxis, right? Anaphylaxis means you're not protected, and it actually can lead to something called anaphylactic shock. And as we learned, shock is deadly, right? It's potentially life-threatening. And so here now, all of a sudden, rather than a localized release of your inflammation mediators, the inflammation mediator circulates the whole body, right? And all of a sudden now it's like this mass drop in or blockage of airways as things start to get really constricted. Your breathing starts to get constricted, right? But your blood vessels start to open up. Blood pressure can drop, and then that can lead to shock, right? And so it really depends on the level of hypersensitivity that you have, right? And so for some of us, you have some sort of food allergy, and then it's kind of irritating, right? Some of us have respiratory issues that's kind of irritating. Some of us, if you breathe the wrong thing, you go into serious, absolutely cannot breathe, you might die. Some of us eat a peanut and then you get these huge hives, everything constricts, you might die, right? So really then it depends. So generally, things like inhalation will lead to localized type 1 hypersensitivity, right? But you can get anaphylaxis, right, through some sort of systemic reaction, right? Typically, that is not. That's when the allergen makes its way to the bloodstream, right? And same with the ingestion, right? So you can have your food or drug allergens, typically localized. Sometimes it can get worse. Okay, so that's type 1 hypersensitivity. Super quick review. Inflammation response due to IgE, right? Sensitization. IgE gets released, coats mast cells, right? Subsequent exposure, the allergen binds to our IgE, triggers the mast cell response, which releases inflammation mediators, right? And then typical response is localized, right? But it can go systemic. Okay, so that's type 1. Let's talk about type 2. Type 2 is sometimes called cytotoxic. And remember, we defined cytotoxic before. Cyto for cell, toxic for poisonous, right? So these are now hypersensitivity attacks on cells. So the ultimate outcome, not goal per se, is to attack cells. And this happens when IgG and IgM bind to antigens on our cell surface, right? So we have some sort of cell coming in, IgG, IgM binds to it, and then we have that attack, okay? And so here, the binding will result in complement activation, right? As we saw, it can lead to the whole membrane attack channels and that kind of stuff, leading to cytolysis, right? Or ADCC. Do you guys remember what ADCC stands for? It's a really long term, but I'll start it for you as antibody. Antibody, dependent cell-mediated cytotoxicity, right? So basically we have complement activation that kills cells, or we have T cells that can come and kill cells, right? And the antigens can be exogenous, right? And this is probably the most common example. Remember, exogenous coming from outside of the body, or endogenous. A little bit more rare, but it definitely can happen. And we'll talk about that as we may not talk about that. I think our two examples are going to be generally exogenous / pseudo-endogenous. But basically what we have is we have our cells, right? These are the antigens that are on the surface. We make our Ig. Hey, what kind of antibody is this? Is this IgG or IgM? this is IgM, right? And you can tell because it's a pentamer, 1, 2, 3, 4, 5, right? So IgM coats, binds to this, and then says, okay, well, we bound this up. These guys are bad. Let's destroy them, right? And so complement gets activated, right? Or natural killer cells or cytotoxic T lymphocytes can come in and then destroy those things. Most common example of a type 2 hypersensitivity is the ABO blood group incompatibility, right? And so the way this works is that our red blood cells, they're generally not the best cells. They don't really do anything except carry oxygen for us. I guess they're super important. But they don't have a lot of surface markers, right? But they do have some. And so these carbohydrates, red blood cells have these carbohydrates on the surface of red blood cells. And this is what we refer to as our blood types, right? And we have 2 carbohydrates, A and B. Okay, so if you're an A blood person, you have the A carbohydrate. If you are a B blood person, you have the B carbohydrate. And then you can actually have both A and B. And then you'll be called AB. And then if you have none of the carbohydrates, then you will be considered O. And this is genetically controlled, right? So we actually have 2 copies, right? Because we are diploid, right? So someone who's an A, so you get one of your alleles, the A or B or none, from your mother, right? The female in your life. And then you get the other allele from the male in your life, right? So you can be AA, which means that you have the A antibody. You can also be AO, right? Which means that you're still A, and then you have an O. Same with B, you can be O. AB, you gotta have one of each, and OO, you gotta have none of each, right? So this is controlled genetically, right? So remember, if you are an A blood person, A is self because you have A blood, right? So what ends up happening over the course of your first few weeks of life is that along the way of B cell maturation, right, we develop these things called isohemagglutinins. And these are antibodies that react with non-self blood. The mechanism is complicated, right? So we're not going to talk about that. Just understand that it happens. So, the first few weeks of life, you actually have no antibodies against any of the blood antigens. But after the first few weeks or so, you get them, and then they persist for life, right? So if you're an A blood, A is self, that means you're going to have anti-B antibodies. Similarly, conversely, if you have B blood, you will have anti-A, right? Because B is self, A is non-self. AB blood, you have none, right? Because A and B are self, right? Therefore, you have none. And O blood, right? A is not self, B is not self. So you're going to have both anti-A and anti-B antibodies. Cool with that? Okay. So this is what happened. This is kind of the situation that you're at, right? So if you receive a transfusion with an incompatible blood type, this can lead to something called a hemolytic transfusion reaction, or simply HTR. And it can be potentially fatal because of the mass amount that you get. And typically, you're going to get a lot of blood, right? So we have a type B. Oh, let's go. Yes, we have type B blood. Hopefully you didn't pay attention to what we saw, right? We have type B blood. What type of antibodies are you going to have? That's right, you're going to have anti-A antibodies, right? So in this case now, we have a scenario with a type B recipient that has anti-A, right? They accidentally get type A blood. And this would have happened a long time ago before we knew this kind of stuff. You go rando person, hey, this person's dying. Let's roll up your sleeve and give this person some blood. And if you get the wrong blood, all of a sudden now, this mass infusion of type A blood will react with the anti-A antibodies. And then that leads to this complement cascade, right? Activation of complement leading to lysis of the red blood cells. As those red blood cells burst, we got this secondary triggering of inflammation and this mass, again, potentially leading to shock, right? Potentially leading to a lot of different problems. Okay, so this happens when you get the incompatible. So a fun game you should do is, if you are type A blood, type B blood, type AB, or type O, figure out who can be your blood donor. So if you're type A, who can you receive blood from? If you're type B, who can you receive blood from, etc., etc., etc. So why don't you hit pause, and then we'll share the answers, okay? Okay, so hopefully you hit pause. Oh, I really hope you didn't just play and be lame, right? So this is, by the way, a second chance for you to like do this while I'm stalling. So hit pause and then do this. Okay, so if you're type A, you have anti-B. That means any blood with B you cannot receive, right? So you cannot receive B blood. You cannot receive AB blood because these anti-Bs will react with it. You can receive O blood, and you can receive A blood, right? So I'm going to write it here. You can receive A, you can receive O. Good. Type B blood, you have anti-A antibodies, so that means you cannot receive A, you cannot receive AB, but you can receive B and O. AB, you have no antibodies, so you don't care. You can receive it all. Yay, guess who's AB blood? This guy. So I can receive it all, right? And then O blood, you have anti-A and anti-B. That means, you can only receive other O blood, right? And so that's why, now that we see this, sometimes we refer to O as a universal donor, because, you know, if we're talking about just these blood types, anyone can receive O blood, given the fact that it has no carbohydrates, right? No carbohydrate antigens. And AB is often called the universal recipient because it really doesn't matter, right? There are other blood groups, which we'll talk about in just a second, right? But generally, this is how it goes. So, you know, in an ideal situation, you would match blood types, right? But generally, in an emergent situation, almost everyone just gets O, right? Because you just don't want to risk anything. And then we'll talk about O negative in a second, because that's really what you get. All right, so I kind of teased it. There's a second major red blood cell antigen called the Rh factor. It's called the Rh, for history, it's from a rhesus. It was discovered in a rhesus macaque, and so that's what the Rh stands for, right? Turns out that we have it as well. It's complicated, so we don't want to talk too much about it, just that it's there, right? And so you either have it or you don't. You're Rh positive or Rh negative. And most people, by the way, are Rh positive. Okay, so generally it tends to be pervasive. It's a dominant phenotype, right? Rh factor is a protein antigen, so it's T cell dependent, unlike carbohydrates, right, which are B cell dependent, right? Anti-Rh antibodies then require sensitization. So here now we have another case where there has to be this sensitization to the Rh, and actually, you know, you do get sensitized, and that's why we get these isohemagglutinins, right? But here, the Rh factor that requires sensitization. So you actually get a first, if you get a first dose of Rh positive, if you have the anti-Rh antibodies, right, you get sensitized. So that first exposure causes this slow, generally harmless hemolytic transfusion reaction. Because it's so small and mild, it's the sensitization process. However, subsequent exposure then can lead to fatal HTR. So that's kind of dangerous. And the most common case that this happens, right, is in pregnancies. This is what we call hemolytic disease of the newborn, and this can occur when the Rh negative mother, gives birth to an Rh positive baby, right? So we have a case where we have a mother, first pregnancy sensitizes, right? So we have the fetus, remember they share blood, so the fetus's Rh positive red blood cells enter. The mother now gets sensitized, so it basically reacts to the Rh blood group. Remember, that's non-self, right? Because she's Rh negative. All right, so she thinks Rh positive blood is non-self. So she mounts this very small antibody response. Typically, it occurs late in the third trimester, right? So it happens so late that this small amount of reaction usually is not felt, very unlikely to cause any problems, right? However, she has these antibodies now, right? She's now been sensitized. So she has a second pregnancy with a second Rh negative fetus, right, all of a sudden now these antibodies can attack the fetus, right, resulting in destruction of the fetus, right? Fetus becomes hemolytic, right? So if you, without any intervention, an Rh negative mother can have one Rh positive baby without intervention, right? But because that sensitizes. But upon second pregnancy with an Rh positive fetus, now the mother's immune system will attack that fetus, and then that'll lead to miscarriages, right? As that fetus gets destroyed. So typically, what you do then is you screen mother for a lot of blood work, and there's tons of, anyone who's been pregnant knows they draw your blood like crazy, right? They're like worse than vampires. And so one of the things they do is they check to see whether the mother's Rh negative or not. And if she is Rh negative, they treat it with Rh antibodies, right? And what these Rh antibodies do now is that they bind to the fetus's red blood cells, and essentially they block the mother from seeing the Rh, right? And so the mother will never mount that immune response. And they check it constantly, make sure that that the immune response never happens. In the event that she does get sensitized, well, then they will definitely talk about warnings and whatnots, right? And then again, this treatment usually happens later, I think in the third trimester, because that's when the problem happens. Cool? Okay, so then getting back to the whole blood transfusion, the O negative thing, right? Remember, if you are, so I said I'm AB, right? I can receive any blood, right? In fact, anyone can receive O blood, right? But if you happen to be something negative, AB negative, B negative, A negative, right? That means an O positive blood will actually be bad for you as well, right? Because of the fact that you're receiving this Rh factor when you are negative, right? So again, in the event that you don't know, typically the blood that they use in the emergency room is O negative, or O neg for all you cool people, right? Because it lacks any carbohydrate antigens, the A and B, but it also lacks the Rh factor. And those are the two biggest blood groups that'll affect things like HTR, okay? Again, ideally you would match, so you're not wasting the O neg, right? But the O neg, if you are an O neg person, by the way, they would love it if you can donate blood like all the time, right? At every single interval that you're allowed to give blood, they would love it because your blood is super valuable. Mine, not so much. Okay, that's type 2 hypersensitivities. Antibody attack on cell antigens. Okay, so the antibodies bind to the cells, right? Some sort of cell surface antigen, and then triggers this inflammation, ultimately leading to destruction of the cells. Type 3 hypersensitivity is a little bit wonky, right? It's something called an immune complex. And what an immune complex is a bunch of antibodies and a small amount of antigen. So it typically happens when there's much more antibody to antigen. Okay? It's kind of a strange situation. And what you end up getting is these clumps, right? In this case, they're showing IgG as an example. IgG is binding to the antigen. You form these large clumps or aggregates, right? And this is an immune complex. And it's always going to form against soluble or free-floating antigens, right? And this can be, sometimes it's a mechanism for things like penicillin sensitivity, right? So if you're allergic to penicillin, it's probably a type 3 hypersensitivity. Any type of injectable drug, right? Things that are floating around, just because you happen to have some sort of antibody that sticks to it. Okay, so that's typically the most common case. And then there are other cases as well. And we'll see actually with some autoimmune disorders as well, but we'll get to that later. And then so what these things then do is, because this doesn't get cleared out, they don't really know what to do with this. It's like in this weird clump that they don't know what to do with. It ends up usually sticking to tissues, right? And so it just kind of sticks to tissues, kind of binds up against them. And then, well, now our immune system says, hey, look, there's a clump of antibodies. Clearly it's bad, so it binds to it, right? Binds to the FC region, bringing in things like macrophages, right? And then you just get this attack, this inflammation attack to try to get rid of that, right? And ultimately what ends up happening is that it's destroys the system, right? Destroys the tissue that it's stuck on, right? the reaction can be localized, so you can actually have this like local reaction. So wherever you happen to get an injection, for example, you can get this localized reaction, or if it goes throughout, you can get a systemic reaction, and that's called serum sickness, right? So serum sickness is simply a type 3 hypersensitivity that is, rather than localized to one specific tissue or area of the body, it has now spread everywhere, okay? I don't really want to say too much more than that, right? Again, it's kind of a rare, semi-complex situation, right? But we'll just kind of leave it at that, right? Immune complex, right? So type 3 hypersensitivities are immune complex reactions, which are simply aggregates or clumps of antibody and antigen. It binds to soluble antigens, right? Stuff that's free floating, and then sticks to tissues, and then this inflammation response ends up damaging the tissue, right? It can be localized, so typically if it's something like a drug sensitivity, you'll get it at the site of injection, or it can be systemic, and we call that serum sickness because it's just all over your body because it's circulating in the blood. Type 3. Okay, type 1, type 2, type 3 are all antibody dependent, right? All antibody mediated, so its antibodies are very much involved in this process. Type 4 is cell mediated, right? So it's regulated by T cells, not antibodies. You can subcategorize type 4 hypersensitivity, so you can have type 4 subcategory 1, type 4 subcategory 2, type 4 subcategory 3, okay? And then the general categories are really dependent on the type of antigen, so whether it's a soluble antigen or whether it's cell associated, and then the type of cell, the effector cell that's been activated, right? Whether it's macrophages, things like eosinophils or CTLs. Okay, and so we'll talk about some examples of these, and we are all familiar with an example of this, and I will mention that very soon. I'll probably come back to this slide. Okay, so again, it's complex, so we're just going to keep it simple, right? I like to keep all this stuff simple just so we can understand what's going on, right? But like with everything, sensitization, right? So first exposure leads to sensitization. In this case, it's actually very similar to what happens in a type 1, right? You get exposed to the antigen or the allergen, right? And then the antigen presenting cell picks it up, right? And then presents it, in this case now, to a T cell. Right? T cell gets activated, memory proliferates, and then it becomes the memory T cell. Right? So we have our memory helper T cell in this case. Upon subsequent exposure, the memory helper T cell then becomes activated, releases cytokines, and starts telling, hey, macrophage, you do this. Hey, cytotoxic T cell, you do this, and starts attacking this allergen, right? Leading to the response. And again, the response really depends on where it's happening within the body. Okay, so once again, sensitization, first exposure. So if you've ever been stuck by poison ivy, first exposure usually doesn't cause anything. It can be a little prickly, right? But it's not a big deal. Subsequent exposure causes it to be extremely painful. You get these crazy lesions like this person has. So I know I've been exposed to poison ivy at least once. And I'm always trying to remember, did I get like a hardcore sensitivity or not? So I'm not even sure. I know I'm at least sensitized. I don't know if I had a reaction. I definitely didn't get this crazy lesion that you're seeing in this picture, I think. But I don't remember. All right, so most of us have probably never seen poison ivy because it doesn't exist here, right? But we all have seen this. Our tuberculin reaction, right? So when you get your TB test, this is what happens, right? And so it's a kind of an indirect measure of whether you've been exposed to Mycobacterium tuberculosis, right? And so when Mycobacterium infects you, if you have tuberculosis, it primes the macrophages, right? So the macrophages, it actually infects the macrophages, and then the macrophages are able to present the antigens, right? Activates its T cells. T cells are now, we have our memory T cells against tuberculosis, right? And it actually goes latent, right? So now we have this case where you've been infected with Mycobacterium tuberculosis, right? Your body has seen it before, but you don't have active infection, right? But your macrophages have been primed. So when they stick the little thing under you, what they do is they stick in the tuberculin. It's a protein from Mycobacterium tuberculosis that's immunogenic, right? And so when they stick it in you, what they're looking for is this hypersensitivity reaction, right? So, and it's a localized reaction, right? And so if you have seen, or if you are infected currently with Mycobacterium tuberculosis, your T cells are going to react against that tuberculin, and then you can see that real red puffiness right at the site of injection, right? So real red puffiness indicates that you've been exposed to Mycobacterium. That means you are likely infected with tuberculosis. Now, and then of course, no reaction means you've never been primed to it, okay? Now, the thing is, for some people, this causes you can have a response to it even though you're not infected. So I actually received the BCG tuberculosis vaccine, I think. I don't know. That's what my mom told me. So whenever I get the tuberculin skin test, I do get a small reaction, right? It's not large enough for it to say, oh, you've been sensitized. And sometimes I'll see it, the nurse looks at it and then puts, measures it because they have to take the size, and they'll write down like 2 millimeters or whatever, right? Sometimes she just looks at it and says, it's not big enough, just put 0. Because I think if they put a small number, then maybe they say, okay, maybe this person needs a chest x-ray, right? But so that's what they're doing. They're sticking a little bit of the tuberculosis protein in there, and then they're looking to see if the T cells attack it. And so that would be the localized response, okay? Okay, those are hypersensitivities, right? There's a wonderful chart table in your book that goes over the type 1, 2, 3, and 4 hypersensitivities, right? The what is causing it, what is mediated, right? Whether it's things like IgE, IgG, IgM, or T cells, right? What the antigen looks like, what the mechanism of activation is, and then just some examples, right? So this is a really nice summary of it, right? If you're not much of a reader, though, I would go back and just look at the slides. And you got to have it organized, right? Figure out how can you organize these things so then that you know the difference and you can recognize the difference between type 1, 2, 3, and 4. Okay, so look for the keywords, look for the things, and associate it. So don't memorize keywords. If you're just sitting there memorizing immune complex, right, and then on an exam you see, oh, there's immune complex, that must be the right answer. It's in my notes, right? But if you don't know for which hypersensitivity it relates to, well, then you're pretty much just memorizing random words, right? So we definitely don't want to do that. Okay, now's a good time, I think, to take a break. We're about half an hour in. So take a break, and then let's come back and we'll talk about how we diagnose hypersensitivities, how we treat them, before we move on to other issues with the immune system. So if you took a break, welcome back, right? If you've ever had an allergy test, right, this is typically the way we test for type 1 hypersensitivities, right? It's called a prick puncture skin test, where they take these needles that are loaded up with rando allergens, right? So I say rando, right? But really, based on what you say, the time of year that you get whatever your issues may be, right? And then, of course, whatever your specific issues are, they will kind of load it up with what they think you might have issues with, right? So if you're talking about, of course, like, oh, I think I always break out when I eat certain types of food, they might put food allergens on there. If you say, oh, I have like respiratory issues, they might put various common respiratory allergens in there, and they're basically just picking underneath, right? Less common is the intradermal, where they stick it underneath, right? It's a little bit more invasive, right? But it's most common is this prick puncture skin test. And then what they're looking for now is this small inflammation response, right? It's called a wheal flare, right? So the wheal is like this raised bump. I mean, you can tell, right? You get stuck and you're like, whoa, this one thing is like way different than everything else, right? So you can tell. And then the flares is redness that extends from the wheal, okay? And then really the trick in is, what the heck is number 3, right? So that's what that is, right? And then they'll have some controls as well. All right, so you see a wheal flare, it's positive. So this is how we diagnose type 1s. We can also diagnose type 3, but because there are so many different causes for type 3 sensitivities, hypersensitivities, it's complicated, right? So it's a complicated analysis, right? It's something that requires really looking into cells and blah, 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 right? So we'll just leave it at that. We'll talk about the prick puncture skin test for type 1 hypersensitivities. Cool. All right. So you have now been diagnosed how do we treat it? Well, to be honest, if it's like a minor annoyance, they say just take your antihistamines or your anti-allergy medicines, right? Your hay fever medicines. We always say allergy medicine, but that's such a general term, right? Really, it's this anti-inflammatory against usually respiratory type of allergens, right? But it can be used against anything that you used to treat anything where you have an improper inflammation response, right? If it's serious enough where it's actually affecting your life, right? You have such serious respiratory issues, right? Then what they can do is they can try to desensitize. It doesn't have the greatest success. No, wait, that's not true. It's about 50-50 from my understanding, right? And the idea here is that they give you repeated injections of this allergen, right? The thing that you're sensitive to. And the idea is that, it's a small, low dose. And then I think it's on a monthly or maybe every 2 weeks, or it might even change as you go further into treatment. My old supervisor, he used to get his monthly allergy shots because it was really bad for him, right? So it's this extremely low dose, and they gradually increase the amount of allergen that they put in. And the idea here is that they want to induce an IgG response. And what IgG does is it blocks the allergen, right? Instead of IgE, which triggers the immune response, right? So it essentially neutralizes the allergen before it gets to your mast cells, right? And because of the fact it's this sort of race between IgG versus IgE, and it only takes a small amount of the allergen to bind, it doesn't have the best success rate, because you can have these IgGs, but if they're not getting to the allergen before it triggers the inflammation response, you can still get your signs and symptoms of your allergies, right? However, it's not bad, right? It's something that definitely can help in this response, right? And it has also worked against some food allergies, right? So if you have certain food allergies, again, usually if it's not fatal, right, they can gradually increase the amount of the food that you are allergic to, and then slowly work it back in such that you're creating IgG. And now I have had friends who tell me that they're allergic to dairy, or they're allergic to shrimp, or to whatever, but they love it, right? So what they always say is like, I'm just gonna have a tiny bit. I know a tiny bit. Maybe I'll break out a little bit, right? I'm just gonna have a tiny bit. And they say that over the course of years of doing that, they've actually really reduced the reaction they've had to the foods, right? Do I recommend this? Yeah, probably not. But again, if it's just a mildly itchy hives kind of stuff, then go ahead and goof around with it, right? But if it's like, this one peanut can kill me, or it's smelling a peanut can kill me, I definitely would not try to desensitize yourself, right? But again, that would be the mechanism of those that try that. Generally, like I said, if you have mild allergies, right, you just treat it with anti-inflammatory drugs. It would go same with type 4 hypersensitivities. So things like rashes due to contact dermatitis and whatnot. They'll say, well, inflammation is a problem. Let's just treat it with anti-inflammatories. Doesn't treat the root of the problem, by the way. It only treats the symptoms, right? But because you can't go in there and just remove the T cells that are causing the problem, that's kind of how it is. And then if you get anaphylaxis, where you get this mass, huge systemic inflammation, epinephrine or adrenaline shots counteract that, right? So those of you who have severe life-threatening allergies, right? You have your EpiPen, Epi for epinephrine, and just inject yourself, and then that helps counteract it long enough. By the way, you don't just inject yourself and they say, okay, I'm done, I can go back and watch my TV show, right? And you say, this lets you survive long enough to get yourself some proper medical care so you don't drop dead, right? So that's general treatment there, but really, this is the best one. Avoid allergens, right? So, those of you with intense, deadly allergies, you avoid them. Don't eat the certain foods. You always have to ask, right? And then for some, if it's like hay fever, move, right? Some people have to move because it's so bad, right? So they move to areas of the world where whatever's giving them hay fever doesn't cause them any problems. So then now I'm going to soapbox a little bit and say that hypersensitivities are dangerous and deadly, right? So, if you go to a restaurant, for example, and say that, I can't eat gluten. Can you make sure this is gluten-free? But you're doing that because, you think it's quote unquote healthy, right? That's different from those that have gluten sensitivities who, gluten can cause some serious issues, right? So, make sure that you understand the the lengths that people have to go to, right? So restaurants that are allergy friendly, you know, they will go through great lengths to prepare food that is peanut free, gluten free, whatever free, because of the fact that it could kill them, right? But if you're just like, oh, I just want to avoid the gluten, right? That it kind of pisses them off, right? So just make sure that you are not confusing your lack of desire to eat something versus this something can kill you, okay? And then you will definitely make the restaurant people much happier. And of course, in your own home, you can create the food however you like. All right, so these are hypersensitivities. These are responses, unwanted responses to allergens or antigens that are generally considered safe, okay? Sometimes the unwanted immune response isn't against a foreign antigen, it's against yourself, right? And so remember this whole idea of self, right? Something we call tolerance. Normal, healthy immune system does not attack self, right? And we talked about B cell maturation, T cell maturation. Along the way, the B cells and T cells that attack self are eliminated, right? It's all part of this maturation. It's very complicated, very involved, right? And because theoretically, our B cells and T cells can interact with any single antigen in the world ever, theoretically, right? Which of course includes us, right? And the way that they are able to do this is through this random assortment, random creation of antibodies, random creation of T cell receptors. So those that interact with self are eliminated, right? And so that's called tolerance. Autoimmune diseases are when there's a loss of self-tolerance, right? So sometimes there can be various conditions that lead to loss of self-tolerance. And some hypersensitivities are mechanisms for autoimmune diseases. So some hypersensitivities will eventually train our immune system to start attacking ourselves. Autoimmune disorders are so poorly understood, right? It's complicated, right? So sometimes it's genetics, sometimes it's actually easy, sometimes it's the environment, sometimes it's a combination of things, right? So you don't really know. There's one type of T cell, the regulatory T cells, the Th17, which we talked about last module. For a long time, they didn't really know what these T cells did, right? But now they know that they're, at least in some part, at least one of their main jobs is to help maintain tolerance. And they do that by getting rid of self-reacting T cells. How it does that isn't super clear. And if there are any other roles that a reg T cell does, or sometimes called Treg, if you want to be all cool, right? How it does that, not fully understood, right? But there's definitely some sort of process that is maintained to help loss of self-tolerance after our T cells and B cells have matured, right? And then ultimately, when you do have some sort of autoimmune disorder, it's going to be some sort of damage to tissue as a result of inflammation. There's always inflammation, right? It's such a bad thing, right? Again, good when it's good, bad when it's bad, right? And so generally the treatment for autoimmune disorders are going to be things like immunosuppressive drugs, right? Which help tamp down your immune system, which of course has a secondary effect that makes you a little bit more prone to other diseases, right? Because your immune system has been squashed down a little bit, right? Sidebar, some of those autoimmune disorders like rheumatoid arthritis, plaque psoriasis, you see the commercials for, and they always say, don't take it if you have TB or something like that, right? Well, because, your immune system kind of keeps TB if you have a latent infection, keeps it down. If you take these immunosuppressive drugs, like for rheumatoid arthritis or plaque psoriasis, maybe you'll wake it up and get TB, which is worse than plaque psoriasis, right? Or you can use steroids, which are anti-inflammatories, again, to work on keeping the inflammation down. Autoimmune disorders can be classified into two large groups: organ-specific, right, where the autoimmune disease affects one specific organ or tissue. And so we'll talk about, for autoimmune disorders, we'll talk about a few diseases and some examples. We'll talk about some examples of a few diseases and just kind of highlight some of the things. There are more examples in your book. You're welcome to read them. Really interesting stuff, right? And but we'll talk about a couple of organ-specific ones, specifically celiac disease and type 1 diabetes. And these are things that we hear about all the time, right? So celiac disease is specific to the small intestine, right? And this is a reaction to protein in grains, right? So it's typically gluten, but it can be other proteins within grains, right? And so those that they have to go gluten-free is because their exposure leads to these antibodies that now start to attack the small intestine, right? So it binds to the small intestine, right? Causes this inflammation, pain, diarrhea. It's extremely painful. It's not something that's pleasant, right? So these people have to avoid gluten and generally actually have to avoid grains because of the fact that it can cause this type of response, okay? How it happens, a little bit unclear, it is complicated, right? But basically, your body has now reacted negatively to the grains by attacking yourself. Kind of a strange thing, right? Type 1 diabetes, right? So diabetes in general means an inability to deal with the glucose in the blood. And there are many types of diabetes, right? In type 1 diabetes, this is an autoimmune disease where your body attacks the pancreas. And more specifically, it attacks the beta cells of the islets of Langerhans, which are the insulin secreting cells, right? So these are the cells that secrete insulin, right? So when your immune system starts attacking these cells, all of a sudden you don't produce insulin anymore, right? And so this is treated with regular insulin shots. Typically, type 1 diabetes is called juvenile diabetes because, remember, sensitization, right? So your body at some point decides that these beta cells are bad, right? Gets sensitized, has to mount this response. And then once it gets sensitized and is mounting this response, it's this slow, gradual thing that eventually manifests as a child or young adult. It's very rare for you to go your lifetime and all of a sudden you're 40-something and says, oh, you got type 1 diabetes, right? Different from type 2 diabetes, which is lifestyle, right? It's just due to the diet that you have, somewhat genetics as well, and the way you process things, right? Type 1 diabetes is an autoimmune disease, okay? Okay. So these are organ-specific autoimmune diseases, right? We can also have systemic, well, that's what AD stands for, by the way, if you didn't know. We can also have systemic autoimmune diseases, right? And these are target multiple organs or tissues, right? And so we'll talk about three, specifically multiple sclerosis, rheumatoid arthritis, and lupus. Multiple sclerosis is an attack on the central nervous system. And basically what happens here is that the immune cells have figured out a way to cross the blood-brain barrier. And then they start attacking elements of your central nervous system. And it ends up creating these lesions, right? And that's kind of where this name multiple sclerosis comes from, right? These lesions that are formed along the central nervous system. And then so the outcome is going to be some sort of disruption in signaling, right? So, anything that requires neuron signaling, so your muscles, for example, right? And then generally you're going to get muscle weakness, poor balance, you get numbness, plus many other things that come along with it, right? So these are just some of the main ones. Right, but basically it's, your immune cells are hanging out in areas they really shouldn't be, right? Rheumatoid arthritis, very common autoimmune disease, right? And again, arthritis in general can have many, many causes, right? Rheumatoid arthritis specifically is an autoimmune disease where T cells are starting to attack your joints, right? This is an example where there's a type 3 hypersensitivity reaction. So something happened, you get type 3 hypersensitivity. These immune complexes, remember immune complex mediated, right? Kind of hang out in the joints and all of a sudden the T cell starts attacking them, right? And because you can't get rid of them, it's just kind of there, right? And it's this chronic inflammation in joints, right? And so typically this happens over a lifetime. And so here the treatment would be some sort of usually immune suppressant. Lupus is really strange one. It's when your body starts to attack nuclear proteins, it attacks your own DNA, right? What's up with that, right? And again, this is this then induces a type 3 hypersensitivity, and the attack is generally on many cell types. The overall response for those who have lupus is going to be severe fatigue. People will go through these episodes where they're just so fatigued, right? Because your body is mounting this systemic immune system response, right? Fever, and then this characteristic rash on the face. I don't know how well this shows up here. I think you see it in your book as well, but they call it a butterfly rash, where it rashes like this, right? But it's also sometimes called a wolf mask, right? And that's where the name lupus comes from. And on the left here, we have a poor fella with rheumatoid arthritis, where it's so bad now that they just the joints are absolutely so inflamed that he cannot move his fingers very well. And so they're almost stuck in this position. So that's bad. This is sometimes what happens when the immune system goes a little haywire. And again, it's not necessarily a stronger immune system per se. It's just a misguided, misdirected immune system. One that is attacking things we definitely don't want it to attack. We are maybe 2/3 done. So if you want to take a break now, you can take a break. Otherwise, we have about another third left, right? So if you don't want to take a break, let's just keep on going. And we'll talk about sometimes when the immune system is doing what it's supposed to do, but we don't want it to. Okay? And so this is in the case of things like organ transplants, right? And so let's define organ transplants, right? Grafts are basically a transplantation of an organ or tissue to different locations. And so you can have many different types of grafts, right? An allograft is one where the recipient is genetically different from a donor. This is the most common type of transplant that you're going to get, maybe. An isograft is one where the recipient is genetically identical from the donor. So this is only happens in the case of identical twins because they are the only people, as far as we know, that are genetically identical. An autograft is where the recipient and donor is the same person. So, I'm going to change what I said. This is probably the most common type of graft. And this happens maybe if you have some sort of, maybe a burn or some sort of severe issue in one part of the body. They'll take skin from one part and then use that as a graft for the other part. So I actually have no numbers. I'm just saying things off the top of my head. But I'm just going to say this is the most common type of graft that'll happen. And then a xenograft is when the donor's an animal. Sometimes, and I think you've heard of this, where you can get a pig heart. And it's almost always pig because their organs are very similar to human. Has been transplanted into a person, right? So those are possible as well. Okay, so we of course have to do transplants because in cases where you have organ failure, right? Or in the case of skin grafts, you have some major skin damage and it's just impossible to heal. So we then just move some small parts of your skin to another part, right? And again, especially for allografts and xenografts, right? And allografts out of anyone where there's a different donor, allografts are going to be the most common there. You got to think, what's going to happen? Well, you're taking cells from another person, an organ, not just cells, right? These are non-self. So what should your body do? Your body should mount an immune response. It is the right thing to do. However, we don't want that, right? So, there's always going to be some sort of risk of rejection, right? So, if you're talking about the different types of grafts and autograft, there's no rejection concerns, right? Unless there's some sort of goof, it's you yourself, right? And isograft, there's very, very little concern for rejection because they are genetically identical, right? Now, again, it's a little bit more complicated. There are certain things that may affect the grafts, right? But pretty much the same cells, same tissues, same antigens, right? So in an isograft, you're going to have very little concern, right? It's the allografts and the xenografts, because they're non-self, the rejection is possible, right? So we'll talk a little bit about why rejection happens, but we should know, and some of the things that we can do about it, okay? So how do we know, right? Do you guys remember how our immune system cells recognize self? Right? What is that molecule called? Who's got it? Now you're gonna have to dig way in the back of your brain. Anyone? My wife hates it when I do that. MHC molecules, right? So remember, that's how we recognize self. So we have MHC1, MHC2. Collectively, they're called HLAs. And then so you can go back and review this in chapter 18.2 in our last molecule. Last module, right? So these HLAs then they're used to present antigens to T cells, right? And also ways to recognize self, right? Because remember, they are going to always present self protein, right? Sometimes dendritic cells will see a foreign HLA come in and they say, ho, you are an HLA, but you're not one of us. You don't belong here. So the dendritic cell will process that, present that to T cells and say, hey, here are other cells with HLAs that don't belong, right? And then so then the T cells will now start attacking the cells with these HLAs. It's technically, the mechanism is like a type 4 hypersensitivity. Oh shoot, I need to fix that. I'm going to fix that. So if you're going to read the non-narrated PowerPoint, that'll be fixed. But right now, we will just deal with that. So it's technically a type 4 hypersensitivity, right? A mechanism, but it's not really a hypersensitivity because remember, the immune system is just doing what it's supposed to do, right? So how do we deal with this? Well, we always talk about matches, right? Oh, so-and-so is a match. Well, what does that even mean, right? It turns out that there are three genes, right, for the HLAs. So, and then each gene we have two alleles, right? So two copies, right, with many alleles. So we have technically 6 genes that we have to match, right? And so it's this random combination of having the same 2 alleles for each of the 3 genes, right? And so, and that, probability means it's really difficult to find. We are a really nice, genetically diverse population, right? So it's really hard to match, but it is possible. And then, so those are the main ones. There are other ones as well that affect it, right? Generally, families tend to be better matches because you're kind of mixing that same pool, right? So that's where they look first. But you can get random matches from other people, right? So it happens. But again, finding the match is a little bit more complicated than that, right? So like we always say, it's complicated, right? But the closer you can match your tissue to the recipient, the less likely rejection will happen. Regardless, you're always going to have some sort of weird goofiness when you first get reaction, right? So almost always, no matter what or who the recipient, the donor is, the recipient will always be under some sort of immunosuppressant therapy just to prevent kicking the graft rejection, right? However, that can lead to many complications, right? Because of the fact that, you are on immunosuppressants, so then you are more prone to other infections because your immune system isn't working well. Right, and so this is then one of those things that when you first receive your transplant, remember sensitization, right? You're going to be initially on immunosuppressants, right? So typically you don't often see rejection immediately, unless it's like some sort of really wacky type of tissue that maybe doesn't like it all, right? But generally you receive your tissue and then, hopefully first things first, is your new organ working, right? And if it is, great, you're going to live for at least a few more months, right? And then they need to figure out, okay, are you going to reject this thing? So they monitor you, make sure and see what happens, right? And then hope that your body doesn't do what it's supposed to do and reject because the match was as close as possible. There's this really interesting thing that happens in certain grafts, right? This is called a graft versus host disease. And this is a neat scenario, because, it would suck if it happens to you, but neat in the sense that sometimes the transplanted tissue actually attacks the recipient. And this happens in things like bone marrow transplant, right? Because remember, what is born in the bone marrow? Our blood cells, right? Including our white blood cells, right? So sometimes when you transplant a bone marrow, with it, you're going to bring a whole bunch of white blood cells, right? And so now we have these donor white blood cells. Now imagine that you're like living in one person and then you get transplanted. You're like, oh, and you end up in this new person and you kind of peek out the door like, where are we? What's going on? And you start circulating. It's like, hey, look at all these non-self things, right? And they start attacking it, right? And then it's essentially now these very overzealous new immune cells recognize the host as non-self, right? Because even though it's like that whole weird, are you the non-self or are you the non-self, right? Really, the new immune cells are non-self, but they're just doing what they're supposed to do, right? And so they're going to start attacking the new host. And then there's this, again, the result will be this major inflammation, cytokine storm, yada, 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 right? So once again, matching is important. The more closely your bone marrow is matched, the less likely this will happen, right? And another thing that that is often done is you try to process it such that you remove as many of the antigen-presenting cells and the T cells as possible, right? Now, even though the bone marrow will have with it the hematopoietic stem cells that it will eventually become blood cells, right? They have to mature before they circulate, right? So most of these will actually get eliminated through the maturation process, right? But it is possible that something slips through and then they cause some problems in the future, right? But generally, match is important, right? Always important. So we have successfully done transplants for quite some time, since 1954, in fact. I didn't fact check that, I just got that from your book, right? And so there have been many examples of successful transplants, many examples of unsuccessful ones, right? So, of course, as medicine progresses, as we get better with technology, you know, what are some of the things that are being done to try to increase the success rate of transplants, right? And so remember, the problem here is recognition of non-self, right? So one of the biggest avenues of research is to take the recipient cells and create a desired organ. So I don't know if you've ever heard of these really weird experiments where they take like a mouse's ear and then they grow a new ear in a dish, right? Well, they do that because they want to see if they can take an organ or skin, typically skin was the first thing that they used, and see if they can get those skin cells to, because our skin cells or our ear cells or whatever, they don't normally divide, right? See if they can take those skin cells and then become actively dividing again, and then they'll kind of grow it around some sort of structure, and all of a sudden now you can grow a brand new ear, right? So that's why they do that. And then that has gone on then to maybe take cells from an animal and then see if you can grow a heart from it or a lung from it, right? And are these functional? Are they the right size, et cetera, et cetera, et cetera, right? So that's probably the most exciting avenue, right? I don't think we're there yet, but that's definitely the most exciting. The other one is using donor animals, genetically modified donor animals, right? Like pigs, for example. I just mentioned how pigs have very similar organs, similar size, similar functions, similar connections, right? So we can just like unplug one and put in the other. And then so these pigs will be modified and then bred so then that their tissues don't contain the immunogenic proteins or those antigens that are the cause for transplantation, right? So when you get a new heart, you don't fear that rejection because it lacks the thing that our immune system recognizes, right? Again, it's more complicated than we make it seem, right? But, you know, that's definitely one neat avenue, right? So those are the two large things, and both of these are progressing to the point that, you know, I feel like maybe in your lifetimes, we might see stuff like this happening on a regular basis, right? And so then in the future, you know, transplants will be much, much more successful. Okay, so we've been talking about unwanted reactions. We've been talking about reactions where your immune system attacks you. We've been talking about things where the immune system is doing the right thing, but we don't want it to. Sometimes we have the total opposite, a lack of a proper response, and that's called an immunodeficiency. All right, so basically there's no immune response, right, or a poor immune response. And so all of these things are immunodeficiencies. Two general types, primary immunodeficiencies or secondary immunodeficiency. Sometimes it's called an inherited immunodeficiency because it's genetic in cause, right? Or acquired, right? Some sort of lifestyle event has caused this immunodeficiency, okay? And for secondary immunodeficiency, malnutrition is probably the most common. If you're not properly receiving the nutrients, your body is not properly able to have a good immune system. Like with our autoimmune diseases, we'll just talk about some examples of immunodeficiencies and then the outcome and the result, right? So for primary immunodeficiencies, lots of them, right? Again, I didn't fact check this is from your book, but it says there are greater than 250 types, right? And the defects can be in either the innate or the adaptive immune response, right? So it's not necessarily going to be in just one or the other, right? And sometimes it can be both. And then some immunodeficiencies are evident at birth. Some develop a little bit later in life, okay? So we're going to talk about three. One called chronic granulomatomus disease. And these are defects in phagocytes. Remember the granules or granulocytes, right? In this case, these phagocytes or their granules contain those digestive enzymes, right? And these ones basically lack those enzymes, right? So what they have then is this inability to digest bacteria, right? So they have impaired antibacterial activity. So these phagocytes then, lack of proper phagocytes means that it's much harder to clear out bacterial infections, right? And these longer infections can often lead to things like granulomas, right? Where you have these poor white blood cells. They're just like all gathering around these bacteria, and they're like eating it, and they just cannot digest it. It's just like, oh. So they don't know what to do with it. They're just sitting there, right? And you get these massive macrophages kind of sitting on top of each other, right? And so if you don't remember what a granuloma is, go back to chapter 17.5. You'll see a lovely picture of granulomas. Here's a multiple. X-linked A-gamma globulinemia, right? And these are, remember, gamma globulins, right? Or immunoglobulins. Those are antibodies. So in XLA, these are defects in antibody production. Right? And basically what happens is that the B cells are unable to mature, right? So something about the maturation process is deficient, right? And so it doesn't get out. And it's X-linked because it's linked to the X chromosome. So this is more of a male disease, right? Because males only have one X chromosome, females have two, but it can be a female disease depending on, females can also get it depending on the whole who the carrier is, that kind of stuff, right? Because you're unable to make mature B cells, right? That means you can't really make antibodies, which leaves you more vulnerable to extracellular pathogens, right? So your viral response is going to be poor. Your bacterial response is going to be real crap, right? Because your antibodies are very important in fighting bacteria, right? So dependent on just your phagocytes doing what it needs to do. So once again, you're going to have just this really poor ability to clear out infections. And in the ultimate immunodeficiency, the king of immunodeficiencies, I should probably use more of a gender neutral term. The royalty of immunodeficiencies is severe combined immunodeficiency, or SCID, right? And this is a syndrome, right? Because there are many causes to SCID, but these are basically defects in T cell and humoral, so T cell dependent humoral response and cell mediated response, right? So very poor antibody response, very poor T cell response, or maybe no immune response. They don't develop memory cells, like really, really, really bad immune system. Like something as simple as a cold is lights out, right? It can be corrected. It really depends on the cause, right? So, if you're unable to, maybe you don't have the hematopoietic stem cells, right? So you can correct that with a bone marrow transplant, but if it's a maturation issue, that might be a little bit more difficult to fix, right? So it really depends on what the cause is, right? Sometimes it's one single gene, and then so people have used gene therapy to treat this. Sometimes it's just multiple things. And then, in this case, poor kid has to live with it, right? So the most famous example, and this is from your book, is this poor child, famously called the Bubble Boy, right? This is a suit developed by NASA, which with a respirator. It has a filter, and then this poor kid has to live in this bubble essentially, right? And then it has a sterile room, so then that person can live in that room, right? And then be able to eat and do that kind of stuff, right? But unfortunately, this is this poor kid's life. They did eventually receive a bone marrow transplant from his sister, right, to try to fix it once they figured out how to do that kind of stuff. But unfortunately, the sister had a virus that was not detected. And we all have these latent viruses, by the way. That's the Epstein-Barr virus. I'm pretty sure it's in your book. You can read it. And then that ended up killing the kid, right? So it's a very difficult situation to deal with, right? And so this poor kid has a very tough life to live, right? Basically, the teeny tiniest little virulent pathogen, lights out. Those are primary immunodeficiencies, right? Secondary immunodeficiencies have a number of causes, right? So they can be from diseases like diabetes over long term. It can result in immunodeficiencies. HIV infection knocks out helper T cells, right? So HIV, the infection actually leads to an immunodeficiency, right? Hepatitis, because of the fact that it's this long chronic infection, will often lead to a really poor immune system. You have an immunodeficiency. And we talked about malnutrition. And malnutrition has a really bad effect on both innate and adaptive immune responses. You can be on immune suppressive treatments, right? So people who are often undergoing cancer treatments, people who are undergoing transplants, as we mentioned, they're under immune suppressive treatments, right? So they're going to have immunodeficiencies. And then just any illness over a long period of time can often lead to some sort of immunodeficiency. So the good news about this is that if they know the cause and if it can be reversed, well, then you can simply rectify the situation by solving the underlying condition. So if malnutrition, for example, is the problem, just feed the kid and then it will not be a problem anymore, right? Certain things like HIV, there's no real cure for that, so there's not too much you can do about that, right? But, eventually, if immune suppressive treatment is causing you the immunodeficiency, at some point when you stop taking it, if you're lucky enough to come off it, then it won't be a problem anymore. Okay, so that's immune deficiency, right? Immunodeficiencies. The last thing we want to talk about is cancer. All right, it's always a tough subject to talk about. But specifically, we want to talk about cancer and our immune system, right? Because we did talk about how there are certain natural ways that we are able to monitor and deal with cancer cells, right? So let's define cancer. Cancer is simply loss of cell cycle control, right? So it happens when a cell divides rapidly and unregulated, right? And so we don't talk about cell cycle here at all, but cells need permission, right? Think of this as you have to have permits, right? It's like building a house, right? You have to have permits to build a house. And it's not one permit. You need a permit for your electrical. You need a permit for your structure. You need a permit for your foundation. You need a permit for everything. It's same with a cell. They need tons and tons of permits before they're allowed to divide, right? Cancer happens when you decide to not go with those permits. And you go rogue and you build one of those massive, disgusting, ugly houses in your neighborhood, right? So these cells divide unrapidly. When they divide unrapidly, right, they become this abnormal mass of cells called a tumor, right? And part of this is because cells, they have this thing called contact inhibition, right? Where you have, this is like some sort of basement tissue, and then these are the cells that are sitting on top of that. They need contact with their basal tissue in order for it to continue to grow. If it ever leaves and does this, or maybe sits on top of there, because it's lacking contact there, your body says, nope, that's no good, right? Because these guys are unregulated, they can actually now start to grow on top of each other, just like this, right? And this mass of cells is called a tumor. Right? Now, tumor is not cancer. It's just an abnormal mass of cells, right? And in fact, because it's just growth on top of each other, you can actually just cut it out and then you're okay, right? So this is what we call a benign tumor, right? What's considered cancerous is if one of these guys starts to float away and then lands somewhere else, and then now we start to grow another tumor here, and then even worse, it just starts to grow everywhere, right? And so these are what we call malignant tumors, right? And malignant tumors are what's considered cancerous. Traditional cancer treatments use radiation. So what they do is they zap right there, wherever there are cancerous cells, tumorous cells, and cancerous cells. And then chemotherapy. Remember, chemotherapy just means using drugs to kill things, right? Whether it's bacteria, or in this case, there are drugs that selectively target rapidly dividing cells, like cancer cells, right? And so they use that to destroy the cancer cells, but because of the fact that, there are other rapidly dividing cells, right? Like your hair cells, right? Your skin cells, right? So there are many unwanted side effects of things like chemotherapy and then radiation, which is really non-selective. It just destroys, right? So it's a really harsh thing to go through. If you've ever had the misfortune of knowing someone, or you yourself have gone through cancer treatments, it's not something that's very easy for the body to take, right? So modern therapies are looking to the immune system to deal with cancer, right? So generally what we're looking for here is a cell-mediated response, right? We do this naturally, right? Remember, our cytotoxic T lymphocytes, our natural killer cells, can react to non-self, right? Because these cancer cells are producing these tumor antigens, right? And then they can either just be recognized as abnormal, so the natural killer cells can get in there, or cytotoxic T lymphocytes will recognize these tumor antigens right? And then mount a response. So we do this cancer screening all the time. But again, it does sometimes make its way through. Malignant tumors are really hard because some of them suppress immune systems. They're spreading all over the place. It's really hard to get at them. Some cancers have cancerous immune cells, so definitely anything with the bone marrow, things like leukemia, for example. Our immune system doesn't really deal well with that. And then some cancers actually resist killing by our cells, and those things are really hard to deal with, right? And so the latest avenue is to use cancer vaccines, right? And cancer vaccines are essentially using or training our immune system to better fight cancers. And there are two general types, preventive and therapeutic. Preventive cancer vaccines prevent cancers from occurring. And we know that some viruses can lead to cancer. So HPV, human papillomavirus, hepatitis B, they can lead to things like cervical cancer, things like liver cancer. Right? So by vaccinating against those viruses, you greatly reduce your risk of getting those cancers. Right? But even more exciting are these therapeutic vaccines. And these are vaccines that are used to treat patients with cancer. Now, I know we define vaccines as a way to prevent disease, right? Because cancer is so long and chronic, we can actually train our immune system to fight active infection. Okay, so that would be a therapeutic vaccine. Remember, it doesn't work with things like a viral infection because viruses are done within a couple weeks. All right, so generally it's not going to work. But because cancers you have for years, you can train your immune system and then have this immune system attack the cancer. And generally, there are two major mechanisms. Use tumor-specific antigens to stimulate immune system cells that now specifically attack cancer cells, or DNA and maybe even RNA vaccines that are created specific for your tumor antigen. Okay, so I'm going to talk about two therapeutic cancer vaccines that have been approved by the FDA. One is sipuleucel or Provenge. It's a prostate cancer therapy. And the reason that this works is because, 95% of prostate cancers have this, I forget the name of it, prostate-specific protein. All right, so this is a tumor-specific protein. So what they do is really cool, is they take the antigen-presenting cells from you, right? They incubate it with this tumor-specific protein, add a few drugs in there, they activate it, make it essentially mount an immune response in a test tube, right? Now they take these activated immune system cells, these macrophages typically, return it to the patient, and then now these woke immune system cells now can talk to T cells and tell the immune system cell, hey man, you should see what I saw, and then train your T cells to now start attacking this prostate cancer. It has definitely some levels of success. I think is it's 62% greater longevity in life or something like that, right? It's not perfect, but it's definitely a really interesting avenue. This one, I practiced saying this, but I practiced saying it so long ago, I almost forgot. The next cancer vaccine, TVEC, that's the cop-out, right, is talimogene laherparepvec. Yeah. It is used to treat melanomas, right, which is a very invasive skin cancer. And this was approved in 2015. This kind of cancer treatment is actually a virus that infects and lyses tumor cells, right? So it's really neat, and it recognizes the tumor cells that cause melanoma, right? And what it does then is it infects it, stimulates the systemic response, and starts attacking the cells that cause melanoma. Really neat mechanism to this, by the way. Typically, viruses, when they infect, right, they kind of like in a normal cell, they start to use the host cell to replicate, and so they find like this. They have to find like this protein that's in the host cell, and then that kind of activates the virus, and then the virus can now use the host cell to replicate, right? Tumor cells, they're goofy. They don't have that. So the virus comes in, and then because it doesn't have that protein, it doesn't need to wait for that okay to replicate. So it just starts replicating. So it starts replicating and starts knocking out the tumor cells, right? As opposed to regular cells where it needs like that second response, right, before it starts to attack. Right, so this virus then has been essentially trained that if that protein is present, it's not going to do anything. But lack of, oh, sorry, I guess it's been engineered so it loses that control, right? So in normal cells, right, that have that protein, the virus can't replicate because that protein will say, okay, well, you need this other thing, this one thing to replicate, right? But in tumor cells, it's lacking that, right? So it's tumor specific. And then there are a lot of other interesting things in development, DNA and RNA vaccines. Specifically, the more we start to learn about the antigens of tumors, the more we can now start to create vaccines that can help prevent. And the idea really would be, it'd be so cool if they can now say, well, we know that, you know, all breast cancers have this vaccine, all brain cancers have this vaccine, antigen, right? And so we can now get a preventative breast cancer vaccine, right? Really preventative anything vaccine, right? So those are the futures, and so things that we should look forward to. Oh my goodness. So this is the last lecture, of our journey of microbiology. We started all the way back in module 1, where we started learning about, you know, the cell, the biochemistry of the cell, right? The general field of micro, made our way through things like, you know, metabolism, cell structure, genetics, disease, right? Immune system, and now here. So at the conclusion of this, hopefully you'll consider yourself a novice microbiologist. Hopefully you know a little bit more to go and learn more about the microbiology that's around you. Because really, if you open your eyes, you will see that microbiology is everywhere, right? We are all biologists, and in many ways, we're also all microbiologists. So that's it for me. Good luck on Module 12 quiz. Good luck on the final exam, the 4th exam. Good luck in life.